So, dear students, Namaskar. I'm Dr. Nutan Prakash Vishwakarma from Department of Biotechnology, Atma University. So, today I'm here to discuss a very important theoretical portion of PCR Primer Designing. So, in this particular video, I'm going to discuss you that what are the characteristics, what are the different features that a PCR primer should have, and what are the different criteria that you should take in care while you are designing the PCR primer. So, as you know, the polymerase chain reaction, which is widely used, and it is a very important invention in the 20th century, developed by Carey-Mullis. Right? So, in what happened actually in the polymerase chain reaction, there is three steps that you already know: the denaturation. In the denaturation, actually, the genetic material is denatured, and it converting the double stranded DNA molecule into single stranded. In second step, that is known as annealing. The primer anneal to the complementary region of the single stranded molecule, and in the third step, that is extension, the DNA is extended by the action of the DNA polymerase. And all these three steps are temperature sensitive, and the common choice of temperature is 94 degrees centigrade for denaturation, 60 degrees centigrade for annealing, and 70 degrees centigrade for extension. So these values are not fixed, that depends on the type of the primer and type of the template. So these values are not fixed, the timing and the temperature may vary reaction to reaction. All right. So that is exactly the same thing happened here in this case. Here you can see in the denaturation at the 95 degrees centigrade, the double stranded DNA becomes single stranded. And then at the annealing temperature, your primer actually binds with the template. And in the 50 to 60 degrees centigrade, your polymerase attaches to primers and extension takes place around at 72 degrees centigrade. So what actually PCR primers is all about? So a primer is a short synthetic oligonucleotide, means in laboratory actually we synthesize, which is used to initiate amplification of DNA, RNA and PCR. And literally primer means to prime. Prime means initiate or start. In vivo, actually in our cell, the primer is required because the enzyme DNA polymerase has no capacity to initiate DNA replication without any primer. So during this process of molecular photocopying, in vitro amplification also of the target nucleotide sequence is initiated by short complementary oligonucleotide. So while we are trying to amplify DNA outside the cell in the PCR, we also need some primer. So that primer we should design artificially. So these primers are designed to have a sequence which is the reverse complement of the region of template or target DNA to which we wish the primer to anneal. And obviously to make success of your polymerase chain reaction, a good primer is the key. If your primer is wrong, if your primer is bad, definitely you are not going to get the amplification. So what are the primers here? See, here I'm giving you the one example, you will understand. Suppose I have one double stranded DNA. So this is known as template DNA, which I want to copy or multiply, right, or amplify. So here the double stranded DNA, you can find the strand are something 5 prime to 3 prime, and obviously here you can see the 3 prime to 5 prime. So if I want to design a primer, so what I can do, if I want to design, if I this is the region which I want to amplify, right? So what I can do, these are the region. So I can generate the reverse complement of the sequence. So if I this is the double stranded DNA, if I denature it, so this is the separate DNA, this is the one strand, and this is the another strand. So from this one, the negative strand, what I can do, I can design the complement. So complement, how I can design the complement? See, the complement of G is C, the complement of T is A, the complement of C is G, the complement of T is A, likewise the complement of C is this G. So this is the complement. If I want to read this, then how I can read? Obviously, whenever I have to pronounce something, I have to start from the 5 prime. So 5 prime, I can read this C A G A T C C. But in the original strand, what I can read? I can start reading from this way: C C A T G G something. So if you see this one, this primer pair, this primer, you will find this is the reverse complement. First it is complement, then it is reverse complement, right? It is a reverse complement of this particular basis. So primers are basically the reverse complement of the template. So that's how once you design this primer, it will come and bind with this 3' to 5', and you will get the forward strand. Likewise, if you want to design the primer pair number 2, definitely that we have to do because our primers are in pair. So in this 5' to 3', I can design the another one, the reverse complement. So these are the primer pairs. By using that, we can amplify the this result. So primer designing, the important design consideration described below are a key to specific amplification with high yield. Number one, what are the different criteria we should take care of while we are designing the primer? The primer length, that is very important. So it is generally accepted that the optimal length of PCR primer is around 16 to 28 bases. Why it is 16 to 28 bases? Because if you take the length of PCR primer is very less, then specificity will be very less. Because you know that if we take the example of the human genome, the size of the human genome is very big, and you will find the bases. Like if I put the length of the PCR primer is only 80 GCT, so somewhere you will find these repeated regions. So if we increase the size of the PCR primer, the chances of uniqueness will be more, right? That's why to uniquely bind your primer with the template, at least the length of the primer we should keep 16, and the length may goes up also. So range is around 16 to 28. In some books or in some research papers, you will find also the variation is also possible. 14 to 28 you may also find, or 13 to 28 you may also find. So these values are not fixed. It depends which type of amplification you are doing. So for safer side, for the examination point of view, then remember as a 16 to 28 base pair. And primer length determine the specificity. Specificity means on which region it actually binds and significantly affects anything to the template. So if the length of the primer is too short, so specificity will be low and resulting in non-specific amplification. And if your primer length is too high, then it will decrease the template binding efficiency at normal annealing temperature due to the higher probability of forming secondary structures such as the hairpin. So if the length of your primer is more, then in that case what is going to happen, since the your primers are single stranded, so they will make the secondary structure from itself also. So that can make the hairpin, and that will reduce the chances, that will reduce the chances of being matched with the being aligned with the, being bind with the DNA. So that should not be too short nor should be too long. So the range between the 16 to 28 base pair, or you can say 16 to 28 bases only, not base pair. So melting temperature. What are the melting temperature? Melting temperature is the temperature at which one half of the DNA duplex will dissociate to become single stranded and indicate the duplex suitability. So primer with melting temperature in the range of 50 to 58 degrees centigrade, that also may vary paper to paper or books to books. Here I am giving you the range. For ideal primer, the length of the melting temperature should be between 50 to 58 degrees centigrade. That may go down and may go up also in some cases. So the GC content of the sequence gives a fair indication of the primer. If you want to calculate the primer's melting temperature, some formulas are, lot of formulas are there. Many softwares are there which they have their own algorithm to calculate the melting temperature. Few of them I am giving you. Just like if your primer is less than 13 bases, then if you apply this simple formula like Tm is melting temperature equal to A plus T, means how many A and T is there multiplied by 2 plus number of G and C multiplied by 4, you will be able to calculate the melting temperature of a primer. And if your primer's length is more than 13, then this formula you may apply. Then Tm equal to 64.9 plus 41 number of GC minus 16.4 divided by number of ATGC, number of total number of bases. If you apply this formula, you will be able to calculate the melting temperature of any primer if you have the more than 13 bases long. So as I already told you, there is a lot of, I will discuss later on, there is a lot of softwares are available, each one have their own algorithm. So there is a separate formula to calculate the melting temperature, but these are the simple simple formula to calculate the melting temperature. So primary annealing temperature. Primary annealing temperature, the primary melting temperature is the estimate of the DNA hybrid stability if melting and critical in determining the annealing temperature. So on the basis of the melting temperature, you can calculate the annealing temperature. So if too high melting, if annealing temperature is too high, it will produce insufficient primary temperature hybridization, resulting in low PCR product yield. If your annealing temperature of any primer is very high, so definitely it is not going to bind with the template. So you will get lesser primary template hybridization. And if it is too low, then we may possibly lead to non-specific product caused by a high number of base pair mismatches. So this is the very simple formula to calculate the annealing temperature. Ta equal to 0.3 multiplied by Tm of primer plus 0.7 Tm of product minus 14.9. So where Tm equal to melting temperature of the primer, Tm equal to product means melting temperature of the product. So the usually annealing temperature should be the annealing temperature of both the primer pairs, like the forward primer and the reverse primer. There should not be more than 2 to 3 degrees centigrade difference. And the GC content, if you talk about in the primer pair, these are all the things you have to take in care while you are designing the primer. The GC content should be the number of G and C, because you know the bonding between the G and C, there is triple bond, and it is easy to break the A and T because it has only 2 bonds. So if more GCs are there, so definitely that so GC content will going to affect the melting temperature. So GC content should be between the 40 to 60 degrees centigrade. Next criteria is GC clamp. The presence of G or C bases within the last 5 bases from the 3' end of the primer help promote a specific binding at the 3' end due to the stronger bonding of G and C bases. So while you are designing the PCR primer, you try to keep more number of G and C in the 3' position so that it can specifically bind, and it can give you the tight binding with the template. The primary secondary structure. What are the primary secondary structure? Suppose I have designed one primer. So presence of the primer secondary structure produced by intermolecular or intramolecular interaction adversely affect primer template annealing, and thus the amplification, which can lead to poor or no yield of this product. See here, there's the simple suppose this is the simple primer I have designed. I can start reading from this way, like TG A TGC A GC AT. So what happened actually, since the primers are single stranded, so it can make the intramolecular interaction. So here you can say it binds with the G to C, right? So it can make the hairpin like a structure because it is making the bond between this G and C, although it is the single stranded. So hairpin like a structure a primer can make. So if you calculate the strength of this G and C, a lot of formulas are there based on that. Lot of softwares are based on it. So if you see that it is formed by intermolecular interaction within the primer and should be avoided. So you have to choose primer in such a way so that they cannot make the hairpins. But practically it's not possible. So optimally a 3' end hairpin with a delta G of -2 kilocalorie per mole and an internal hairpin with a delta G of -3 kilocalorie per mole is tolerated generally. I already told you there should not be hairpin formation, but ideally it's not possible. So up to -2 kilocalorie per mole or -3 kilocalorie per mole, it is okay, because once you will increase the temperature in the PCR. So in that case, if the primer have made the hairpin structure, it will denature and your primer will be straight. So it will bind with the template. So that is not going to create any kind of the problem. But if it is lot of G and C bonds are there, so in that case, that may create the problem and it will not be specifically bind with your target. So hairpin formation should be avoided. If 2-3 bonds are there, it's okay, no problem. So what the delta G that we have already talked about? So the Gibbs, it is known as Gibbs free energy. So it is the measure of the amount of work that can be extracted from a process operating at a constant pressure. So it is the measure of the exponentially of the spontaneity of the reaction. The stability of the hairpin is commonly represented by its delta G value. The energy required to break the secondary structure. The energy required to break the secondary structure is known as delta G. So larger negative value for delta G indicate a stable, undesirable hairpin. If it is lower one, then it may shows the unstable structure of the hairpin. So presence of hairpin at the 3' end most adversely affect the reaction. Self-timer. Self-timer means once you will put all the reaction mixture in the tube. So there, if two primers pairs are there, if forward primer and forward primer, if forward primer and forward primer may bind with each other, that is known as self-timer. Or two reverse primers may bind with each other, that is known as self-timer. So that also should not be there. Right? Two forward primer and two reverse primer should not make the bonding with each other. But ideally, again, I told you it's not possible practically that no one two forward primer should not make any kind of the bonds or single stranded simple forward primer should not make the hairpin structure. Practically, it's not possible. If you calculate that bonding, if it is lesser than -3 kilocalorie, as I already told you, it's okay. So when primers form intermolecular dimers much more readily than hybridizing to target DNA, they reduce the product yield. Cross dimer. If one primer, like forward primer, binds with the reverse primer, so two separate primers, if they make the dimer like this, that is known as cross dimer. So primer cross dimer are formed by intermolecular interaction between sense and antisense primer. Either you say forward primer or reverse primer, or you say sense and antisense primer, where they are homologous. Optimally, a 3' end crosses dimer with a delta G of -5 kilocalorie per mole and an internal cross dimer with a delta G of -6 kilocalorie per mole is tolerated generally. So it's practically for any primer to follow all these criteria is not possible. So there is some relaxation. All right. Repeats. While you are designing the primer, one thing you also have to take care. Repeat is a dinucleotide occurring many times consecutively and should be avoided because they can misprime. For example, 80, 80, 80 something. So that should not be there. There should not be repetition of the basis. Like here you can see that there is the repetition of 84 times, right? So maximum number of dinucleotide repeat acceptable in any organoid is 4 dinucleotide. More than that should be avoided. Runs. Primer with long run of single bases should generally be avoided as they can misprime. For example, AG, CG, here you can see a lot of time, many times G is repeated. So such type of repetition also should not be there. A maximum number of runs accepted is 4 bases, maximum to maximum, not more than that. 9. The criteria number 9 is 3' and stability. It is the maximum Gibbs free energy value of the 5 bases from the 3' end. An unstable 3' end will result in less false priming. Avoid cross homology. To improve the specificity of the primer, it is necessary to avoid regions of homology. Primer designed for a sequence must not amplify other genes in the mixture. Say for example, I have designed a primer. To amplify any particular region of one gene, that is say for example TP53 gene. So that particular thing should be very specific and that region should not be present throughout the genome. Alright. So suppose you have designed a primer to amplify a particular region of the gene and that gene is again present in some another part of the genome. So definitely you have put the primer to design your desired template, but it may uh, amplify another region. So that should be avoid. Cross homology should not be there. And primary compatibility. Primary compatibility means since you are putting the two primers, forward primer and reverse primer in a same reaction mixture. So ideally, the condition for the denaturation, and annealing is going to be same, right? So your primer should be comfort. I mean, there should not be much more differences in their condition. So there should not be the temperature difference like melting temperature and optimum melting temperature and no the annealing temperature. There should not be more than 3 degrees centigrade difference. Say suppose the forward primer you have calculated the melting temperature as 62 degrees centigrade and reverse you have calculated as a 50 degrees centigrade. So that's not going to work. The maximum difference between that should be between the melting temperature of forward primer and reverse primer, that should be between 3 degrees centigrade. That is the optimum condition, although in certain condition that may vary, that say 4 degree or 5 degree also may also be accepted. So these are the summary that we have already talked about. The uniqueness, ensure correct priming site. Length should be between 18 to 28 bases. You may find 15 to 28 in some literature or 16 to 28, depends type of reactions. And base composition average G plus C, that should be between 50 to 60 degrees centigrade. Avoid long A plus T or G plus C rich region if possible. Annealing temperature between 50 to 65 degrees centigrade are preferred, and ensure that primers as a set have an annealing temperature with 3 degrees centigrade of each other. There should not be more than 3 degrees centigrade difference. And you should minimize the internal secondary structure, like there should not be formation of hairpin and diameter should be avoided. So what is a primer? Is a primer. See here, this is the template you want to amplify. So 5 prime to 3 prime. If this type of tight binding between the template and primer is there, this is the best one. But if in case your primer does not bind in this particular region, but it binds with the 3 prime region, still primer may work, because 3 prime, it binds with the 3 prime, but it may extend the amplification. If such type of condition are there, it is binding with the 5 prime position and 3 prime position, and in between there is some not matching between the, not complementation between the template and primer, still you may get the amplification. But in this particular, if in this particular condition there is the tight binding between the this particular area, and throughout the template and primer it is binding, but at the 3 prime position it is not binding, so definitely you are not going to get your product, right? So this is not a good primer. So a lot of softwares are available over the web, like web-based software like CodeHop, Gene Fisher, Do Primer, Primer 3, Primer Selection, Web Primer, PCR Designer, Primer Pro 3.4, Primo Degenerate 3.4, and PCR Primer Design. A lot of softwares are available free of cost. You can use any one of them. Depends on the type of PCR you want to run. So, and each one of the softwares have their different algorithm. The uh, most of the people use that the primer 3. I will make a separate video that how you can design the primer by using the primer 3. All right, so you can use any one of the software which you like. These are again more softwares like the primer generator, the e-primers, the Primo, the Primer Quest, Math Primer, Raw Primer, Medusa, the Primer Primers project. Standalone software, these are the software which you can download from the over the net and you can install in your PC. And by using these softwares, you can design the primer against any particular template. So these are the long list of softwares which are available. Some of them are free and some of them are paid also. They are the proprietary softwares. You can buy it and you can use it. Again, the Genome Pride, Fast PCR, Oligo 7, Primer Designer, G Prime. Okay, so a lot of softwares are there. So these are the theoretical aspect of primer designing. If you like this particular theory part, you can press the button of like, you can share, you can subscribe, you can comment, and we'll see you in next video, where I'm going to make a video that practically that how you can design the PCR primer by using primer 3 or primer blast. So till then, take care. See you very soon. Take care. Jai Hind.